Okay, let's start it. Hello everyone, I'm Chuo, a second-year PhD student in nutrition, and I'm the host of today's seminar. It's a great pleasure to introduce our student speaker, as well as my friend, Ya Pei. Ya is a PhD candidate in the Department of Nutrition and Food Science here at Texas A&M University. During the past 4 years, she has TA'd for several nutrition courses, and she also works as a research assistant in Dr. Wu's lab. Originally, Ya started her undergraduate study as a food engineer at Tianjin University of Science and Technology in China. She's among the best students and received multiple scholarships 4 years in a row. She continued to study in food science and technology, and her graduate research is mainly about investigating the optimized conditions for extracting biological compounds from drugs and the effects on obesity and insulin resistance in diabetic mice. In 2013, she joined in Dr. Wu's lab, studying the function of adenosine A2A receptors on inflammation and insulin signaling in liver and adipose tissue. As a fourth-year PhD student, she has co-published 6 articles with her lab mates in several scientific journals, including the Journal of Biochemistry, PLOS ONE, and Scientific Reports. I'm so impressed by her accomplishments within the past 4 years, and I'm pretty sure that she will have more papers published upon finishing her PhD degree. Today, she will give us a speech about adenosine A2A receptor, nutritional regulation, and the role in diet-induced inflammation and metabolic disease. Let's welcome Ya.
Thank you very much for the wonderful introduction. I'm very glad to be able to share with you the work that I have done throughout my PhD study. My thesis title is Adenosine A2A Receptor, Nutritional Regulation and Role in Diet-Induced Inflammation and Metabolic Disease. So I would like to start off with some background information. So obesity has been increased steadily in the countries all over the world, and in 2014, there are more than 600 million people are classified as obese, and there are more than 1/3 of U.S. adults are obesity. The health consequence of obesity could be chronic metabolic and inflammatory disease, such as type 2 diabetes, heart disease, stroke, and some certain types of cancers. So people are living in the environments that promote increased intake of unhealthy food and physical inactivity. The unhealthy diet containing the energy-dense, nutrient-poor food that is rich in added sugar and saturated fatty acid, which lead to weight gain. And chronic inflammation is characterized by infiltration of immune cells. The consequence of inflammation is tissue dysfunction. So this research aims at examining the anti-inflammatory effect of A2A receptor on adipose tissue and liver. So obesity is a pro-inflammatory state. Adipose tissue contains a lot of cells, including adipocytes, pre-adipocytes, endothelial cells, and immune cells. Under obesity, adipose tissue will promote the release of free fatty acid, which can serve as the ligand to bind Toll-like receptor 4 and activate the TLR4 cascade. And this can stimulate macrophage to secrete cytokines such as IL-6, IL-1 beta, and TNF-alpha. And in the adipose tissue, there's a lot of macrophage infiltrate, and these cells, with the cells reside in the adipose tissue, taken together can facilitate the production of pro-inflammatory cytokines. And the adipose tissue dysfunction during obesity is characterized by impaired triglyceride storage and increased risk of lipolysis, which can lead to increased concentration of free fatty acids. And the expansion of adipose tissue in obesity could lead to increased macrophage infiltration and increased production of pro-inflammatory cytokines, such as IL-6 and TNF-alpha. And this accompanied with increased secretion of free fatty acids and dysregulated adipokine secretion, such as adiponectin, leptin, resistin, and RBP4. So these adipocytes and macrophage-derived substance taken together can further exacerbate the inflammation of adipose tissue. And in the systemic level, the dysregulated adipokine secretion could cause the liver insulin resistance through ectopic lipid accumulation and inflammation in the liver. The good news is the A2AR, the A2A adenosine receptor, may be one underlying receptor mediating systemic inflammation. The occurrence of adenosine receptors was populated around 40 years ago, and they belong to G protein-coupled receptor family. There are 4 adenosine receptors, A1, A2, A3, and A2 have 2 subtypes, A2A and A2B. And there is research showed that upon activation of A2AR receptor, it can suppress the inflammation in the local injury. So my question is that the activation of A2AR can also suppress the inflammation upon dietary effects. So my central hypothesis is A2AR coordinates metabolic and inflammatory response in adipose tissue and liver in response to diet. And here is some background information about A2A adenosine receptor. The extracellular adenosine comes from 2 sources, and one main source is from extracellular space with the sequential dephosphorylation from ATP with the help of enzymes such as CD39 and CD33. And the CD39 can hydrolyze the ATP or ADP to AMP, and the CD73 can hydrolyze the AMP to adenosine. And the activation of CD39 and CD73 could be hypoxia, inflammation, or adenosine itself. Another source of adenosine is from intracellular space with the help of nucleoside transporter ENT. In the intracellular space, the AMP can be hydrolyzed with the help of 5 nucleotides, and this reaction is reversible depending on the enzyme activity of ADK. And also SAH, as adenosine homocysteine, as adenosine homocysteine can be hydrolyzed to homocysteine and adenosine, and this reaction is also reversible. This picture shows adenosine metabolic pathway inside and outside of the cell. So when there is local hypoxia or inflammatory stimuli, the concentration of adenosine will increase immediately in this local area. And the adenosine will come on over and bind to A2AR, A2A adenosine receptor, and cause the transformation change of this G protein coupled receptor. And the G alpha subunit will leave the G protein coupled receptor and with attached GTP. And as long as the GTP is there, and it will make a bunch of CAMP with the factor enzyme acetylcyclase AC. And one mechanism could be CAMP make pKa protein kinase A more active, and phosphorylated the target protein CREB. And the CREB would decrease the activation of NF-kappa B, which in turn decrease the production of pro-inflammatory cytokines. One mechanism could be the activation of A2A adenosine receptor decrease the production of TNF-alpha by the help of EPC2. And another mechanism is independent both PKA pathway and EPC2, and is the serine phosphatase. And that explains why adenosine receptor A2AR activation can decrease the inflammation upon many stimuli. Here is some background information on the A2AR mediates the anti-inflammatory effects of adenosine. Upon the A2AR activation, the A2AR receptor is required for the uniform formation of granulation tissue and the formation of vascular in a model of wound healing. And there is little research on the adipose tissue regarding to the A2AR activation. And in a recent study, it shows the A2AR activation can increase the activation of brown adipocytes, and as well as increase the activation of browning the white adipocytes, which means the A2AR activation can increase the thermogenesis in the adipose tissue and increase the insulin sensitivity in the adipose tissue. The third one is A2AR activation can enhance the reverse cholesterol transport with the help of macrophage. And this also can help to stop the transformation of macrophage to form cells. The fourth one is in the type 1 diabetic mouse model, the A2AR activation can increase the beta cell proliferation. So the activation of A2AR could be a therapeutic target to treating the type 1 diabetes. And the fifth one is A2AR activation can increase the immunosuppressive effect in ischemia reperfusion injury and allied transplantation. And in the sixth one, in the isolated hypothesis from the rats, the pharmaceutical activation of A2AR can increase the gluconeogenesis in the liver cells. And here is my research. There are four chapters, and the first chapter is the physiological relevance of A2AR in adipose tissue under different nutritional conditions. The second chapter is the physiological relevance of the A2AR in liver under different nutritional conditions. And chapter 3 is direct role of A2AR on adipocytes and macrophages. The last chapter is A2AR protects against overnutrition-induced chronic inflammation and systemic insulin resistance. The chapter 1 to investigate the nutritional regulation of A2AR expression and inflammatory response in the context of obesity and insulin resistance. The wild-type mice on C57 black 6J background were fed with high-fat diet or low-fat diet for 12 weeks, and body weight and food intake were monitored every week. Glucose tolerance test, GTT, and insulin tolerance test were conducted to analyze the systemic glucose level and the insulin sensitivity. In the end, the epidermal white adipose tissue were accessed to measure the A2AR level and the expression and the inflammatory signaling through Western blot and the pro-inflammatory gene expression through real-time PCR. Here is the result. The figure A shows that comparing to the low-fat diet fat mass, high-fat diet fat mass gained much more body weight only after 5 weeks. And after 12 weeks feeding, average body weight in high-fat diet group was around 42 grams, whereas low-fat diet fat mass were only about 30 grams. And the GTT result shows that high-fat diet mass increased their glucose level much higher compared to the low-fat diet fat mass after injection of the glucose solution. and their glucose level keeps high after the injection in 2 hours. And figure C shows the ITT result that showed high-fat mice had increased glucose levels after injection of insulin comparing to the low-fat mice. And figure D showed that high-fat mice have increased visceral fat than low-fat mice in terms of epididymal fat, perinephric fat, and mesenteric fat. And this figure shows that high-fat increased the inflammatory response in the white adipose tissue. And the figure A is the Western blot result. We can see the high-fat mice can increase the phosphorylation of JNK and NF-kappa B, which means that high-fat can have effects on the activation of JNK and NF-kappa B pathway. And the figure B shows that the mRNA level in the high-fat mice of cytokines are much higher compared to the low-fat mice. For example, TNF-alpha, IL-1 beta, IL-6, and MCP1. So my study confirmed that the high-fat can increase the inflammatory response in the white adipose tissue. So how about the abundance of A2AR level in the adipose tissue? The figure A shows the tissue distribution of A2A adenosine receptor. There is a high level of A2AR that found in brain, in immune cells, and in liver and in the white adipose tissue. And the figure B shows that the high-fat mice increase the level of adenosine receptor. And the figure C, in the figure C, I conducted the experiment of real-time PCR. I found the expression of A2AR gene is much higher in the high-fat mice. Summary 1. Compared to the low-fat wild-type mice, high-fat wild-type mice have higher body weight and more visceral fat, and decreased glucose tolerance and insulin sensitivity. And the adipose tissue had increased JNK and NF-kappa B phosphorylation, and increased pro-inflammatory gene expression. And the last but not least, the adipose tissue has increased A2AR levels upon feeding of high-fat. In the chapter 2, I used the wild-type mice fed with low-fat or high-fat for 12 weeks, and the diet-induced difference in A2AR expression in liver was determined through Western blot, and in addition, inflammatory and metabolic biomarkers were tested through Western blot and real-time PCR to compare the low-fat and high-fat treatment. Here in the present study, we also confirmed that the high-fat increased the inflammatory response in the liver tissue. The figure A is the Western blot result. We can see upon high-fat feeding, there is increased phosphorylation of JNK and NF-kappa B in the liver tissues. And figure B shows that high-fat mice have increased the mRNA levels of pro-inflammatory cytokines, such as TNF-alpha, IL-1 beta, and IL-6. And in addition, the figure C shows the liver weight in relative to body weight. The high-fat mice have increased the liver weight comparing to the low-fat mice. And how about A2A level in the liver? We can see from the figure, the high-fat increased the level of adenosine receptor in the liver. Here is the summary 2. The livers of high-fat wild-type mice had increased JNK and NF-kappa B phosphorylation, and increased pro-inflammatory gene expression, and also increased A2AR levels. And in the chapter 3, I want to investigate the direct effects of nutrients on A2AR and the inflammatory response. I used the 3T3L1 adipocytes cultured in low glucose or high glucose medium. and adipocytes were treated with or without palmitate for 24 hours, and cell lysis were used to determine inflammatory signaling and A2AR levels, and the mRNA levels of pro-inflammatory genes and A2AR genes have been analyzed. Figure A shows the Western blot result of adipocytes. We can see that the high glucose and palmitate treatment group had more junk phosphorylation, which means that both high glucose and palmitate treatment had effects on the activation of junk pathway. In addition, Figure B shows that the palmitate treatment will increase the level of pro-inflammatory cytokines, such as TNF-alpha and IL-6. So how about the A2AR level in the adipocytes upon this high glucose and palmitate treatment? So from the Figure A, we can see the palmitate treatment could increase the A2AR level regardless of the low glucose or high glucose treatment. And in the Figure B, we can see the gene expression of A2AR increase significantly in the palmitate treatment group. The second part of Chapter 3 is to investigate the nutritional regulation of A2AR expression and the inflammatory response in the wild-type macrophage. And I isolate the bone marrow from the mice and initiate the outgrowth of macrophage through adding the L929 cell supernatant to the IMDM medium for 8 days. And then the cells were treated with low glucose or high glucose with or without palmitate for 24 hours. And here's the result from the cell lysis of the mRNA. The Figure A is the result of the Western blot. We can see that the high glucose and we can see the palmitate will increase the phosphorylation of NF-kappa B in the wild-type BMDM. And in the Figure B, we can see the palmitate treatment also increased the cytokine expression in the macrophage, such as TNF-alpha. And the Figure B also shows that the expression of A2AR gene is increased significantly in the palmitate treatment group. Chapter 3, the role of A2AR in regulating inflammatory response by comparing with the wild-type mice and the knockout A2AR macrophage. And I used the macrophage to treat with low glucose or high glucose with or without palmitate. And this wild-type macrophage and knockout macrophage cell lysis were used to analyze inflammatory signaling and A2AR level. And here is the result. The Figure A is the result of the A2AR knockout macrophage. We can see the high glucose and palmitate treatment, both of them increase the phosphorylation of junk and NF-kappa B. And the Figure B shows the comparison between the wild-type macrophage with the A2AR knockout macrophage. You can see in both BSA treatment or palmitate treatment, the knockout macrophage increase the phosphorylation of junk significantly comparing to the wild-type macrophage. Summary 3. So upon palmitate treatment, 3T0L1 adipocytes and BMDM had increased junk and NF-kappa B phosphorylation and increased TNF-alpha and IL-6 expression. And the last but not least, they also increased the A2AR levels. Upon high glucose and palmitate treatment, A2AR deficient macrophages had increased junk and NF-kappa B phosphorylation. And comparing to the wild-type macrophage, A2AR deficient macrophage had increased junk phosphorylation. In the chapter 4, I want to investigate if A2AR protects against overnutrition-induced chronic inflammation and insulin resistance. So there are 3 groups of mice: the A2A homozygous knockout mice, A2A heterozygous knockout mice, and the wild-type litter mice. These three groups were fed with heifer diet for 12 weeks, and the body weight and food intake were monitored every week, and GTT and ITT were conducted to analyze the glucose level and the insulin sensitivity. In the end, the livers and the epidermal white adipose tissue were accessed to analyze histology feature and inflammatory signalings and pro-inflammatory gene expression. Here is the result of the male mice. The figure A is the body weight. We can see the heterozygous A2A knockout mice gained much more body weight than the wild-type mice. And the A2AR knockout homozygous mice showed significant increase in body weight comparing to the heterozygous mice. And the figure B, GTT showed that heterozygous mice decreased the glucose tolerance, and their plasma glucose increased significantly comparing to the wild-type mice after ingestion of glucose solution. And the homozygous knockout mice have the severe and bad performance comparing to the heterozygous mice. And in the figure C, it shows that the knockout mice have higher plasma glucose level than wild-type mice after ingestion of insulin solution, and their plasma glucose level keeps high after the injection in 1 hour. This figure shows the female mice, and in the figure A, we can see there's no big difference among the 3 groups of wild-type, heterozygous, or homozygous in female mice. But in figure B, the glucose tolerance test shows that the A2AR global knockout mice has increased glucose level comparing to the wild-type mice. And the homozygous knockout mice has the worst performance in the glucose tolerance test. And the figure C is the ITT result. We can also see the A2AR global knockout mice has increased the glucose level upon insulin injection, and which means the tissues of the A2AR global knockout mice cannot respond to the insulin properly. And here is the result in the liver tissues. And the figure A, we can see in the heterozygous mice, there is much more fat deposition comparing to the wild-type mice. And comparing to the heterozygous mice, the homozygous mice have much more fat deposition than heterozygous mice. And the figure B is the Western blot result. We can see the A2AR global knockout mice has increased the phosphorylation of NF-kappa B. And in the figure C, we can see there is increased the gene expression of pro-inflammatory cytokines in the A2AR global knockout mice, such as TNF-alpha, IL-1 beta, IL-6. In the meantime, the enzymes that related to lipogenesis are also increased in the global knockout mice. For example, ACC, fatty acid synthase, CPT1, and SREBP1C. And the figure D shows that the A2AR global knockout mice have decreased the phosphorylation of AKT. the next one is the result of white adipose tissue. The figure A shows that after 80 staining, and the knockout mice have accumulated much more adipose tissue macrophages than the wild-type mice. And the figure B, we can see there is a switch of phenotype of macrophage from the anti-inflammatory M2 macrophage to pro-inflammatory M1 macrophage. In the figure C, it shows that A2AR knockout has increased the phosphorylation of JNK and NF-kappa B. And in figure C, we can see there is the increase of the production of pro-inflammatory cytokines in the A2AR knockout mice in the adipose tissue, such as TNF-alpha, IL-1 beta, and IL-6. And also there is decreased expression of adiponectin and hormone-sensitive lipase in the A2AR global knockout mice. In the figure E, we can see the A2AR knockout mice have decreased the phosphorylation of AKT in the white adipose tissue. So the summary 4 is comparing to wild-type mice, A2AR deficient mice had higher body weight and decreased glucose tolerance and insulin sensitivity. The adipose tissue and the livers of A2AR deficient mice had increased the JNK and NF-kappa B phosphorylation, increased the pro-inflammatory gene expression, and increased the lipogenesis gene expression. The conclusion: the HEFA diet increased the expression of A2AR in liver and adipose tissue, accompanied with obesity-related inflammation and insulin resistance. It appeared to be a defensive response, which may help protect against inflammatory damage. Number 2, nutrients that had direct effects on A2AR expression in both adipocytes and macrophages, which indicated that A2AR had protection effects on inflammation. So when comparing with HEFA diet wild-type mice, the A2AR deficient mice display a significant increase in the severity of inflammation and insulin resistance. In the last, I would like to thank to all my committee members, Dr. Vika, Dr. Takot, Dr. Wazam, and my PI, Dr. Wu, and also thank for the grant funding source of NIH, and also thank you all my lab mates from my lab. That's all. Thank you.
Questions?
Yeah, I just wondering why you choose the palmitic acid as the ingredient instead of other saturated fatty acids.
Yeah, palmitate fatty acid is the saturated fatty acid. It has 16 carbons and no double bonds. It's the saturated fatty acid. And also is the main source from the HEFA diet, because the HEFA diet, the saturated fatty acid is from lard, the pork fat. And the palmitate fatty acid can bind to the TLR4 and initiate the TLR4 cascade, and for the production, increase the production of cytokines. And also the metabolite of palmitated fatty acid could be harmful, such as the ceramide and the DAG, diacylglyceride. And this metabolite is also harmful to our bodies. So that's why I want to choose palmitate to treat the cells. Yeah. Okay.
Perhaps you can define what you mean by high-fat diet, low-fat diet.
Yeah, I have.
Some additional.
Yes, I have.
Share that information.
Yeah, I have a figure here. So the low-fat diet have 10% calorie of fat, and HEFA diet have 60% calorie as fat. And the main source for the fat for HEFA diet is from lard. And the main source for the carbohydrates for the low-fat diet is the cornstarch. And the low-fat diet can act as the control diet for the high-fat diet. And I have seen from the website of the Research Diet webpage, and the scientists from Research Diet showed that the low-fat diet could be for now the best control diet for the high-fat diet. So because around 30 years ago, we used the Chao diet as the control diet for the high-fat diet, and there's so many variants in the Chao diet. For example, maybe the pesticide in the Chao diet, or the in the flavonoid or from the soybean from the Chao diet. Maybe some variants comparing the low-fat diet. So this low-fat diet could be for now the best control diet for the high-fat diet.
So when you compared the high-fat, low-fat diet in your different experiments with your adenosine receptor knockout or wild-type animals, was there ever a difference in the food intake between these different groups? I might have missed it, but
there's no big difference between the food intake upon the calorie. When you calculate the calorie intake, there is no big difference between the low-fat diet fat mass and high-fat diet fat mass. And the low-fat diet mass, indeed, they contain more of the food. The gram, they have more. But calculating the calorie, they seem to be similar in these two groups.
But so I guess then the obvious question is, and everyone in this field seems to face it, but I don't really see them addressing it. So if they're eating different amount of mass of food, they're eating to a caloric, you're telling me calorically, but the mass is different. What about the micronutrients in the food? Are they not consuming different levels of micronutrients?
Oh, you mean there are carbohydrate, protein, and
no, not macronutrients, micronutrients.
Micronutrients.
You've
Oh.
got a mineral mix and a vitamin mix. Are they consuming different amounts of vitamin and minerals? And does that concern you at all, or not, in terms of the biology you're studying?
Yeah, basically what I'm studying is the knockout mice, and only use the high-fat diet to feed the knockout mice, heterozygous and wild-type mice. I want to see the different response among these three groups after they feeding the same diet, the high-fat diet. Yes.
So could you do something to your diet to accommodate that concern that the micronutrient levels might be different? Did you design the diets?
Yeah, definitely. Yes.
If you could put in different levels of vitamin and mineral mix, and you can back calculate to how much you would have to put in there so they would consume relatively equal amounts once you accommodate the difference in the food intake on the mass basis.
Yes.
There may be an interesting interaction there.
Yeah, definitely. Yeah. Okay, thank you.
And so you said their caloric intake is basically the same. Then why are body weights different?
Oh, it's basically because of the lard in the high-fat diet, and there is around 60% of fat in the low-fat diet, which cause their adipose tissue expansion, and also the fat deposition in the liver from the histology. And when we harvest the animal, it's very obvious. We can see the white dot in the liver tissues. Yeah. And another reason I think maybe is because of the, for example, in the muscle, and there is the increase in the intramyocellular lipids in the muscle cells, and this also could cause the insulin resistance in the tissues of these mice upon feeding the high-fat diet.
And when do you start the diets?
The 5 weeks.
So weaning?
Yes, weaning.
Okay, and so the animals aren't aren't at sexual maturity, but yet you see a sex difference. Have you given any thought to looking at why that is? Have you looked at hormone levels or anything like that in the animals?
Yeah, the majority the work we have done is on the male mice, but also we have done some experiment on the female mice, and there is not so much difference in the body weight among the female mice group. I think maybe one reason is their insulin level in the female mice is different compared to the male mice. Yeah.
And one more, is there a phenotypic difference in the animals and if they're just on a standardized diet, or they're in the knockouts? So when you compare a knockout to a wild type, are there phenotypic differences? Are there things that you could, I mean, without manipulating the diet, or are the knockouts, you didn't describe the knockouts in general, so do they have any reproduction problems or any other issues? Are there any phenotypic obvious things?
Yeah, the global knockout mice, upon feeding the chow diet, there's no big difference on the phenotype. But some specific knockout mice, they may have be harmful or even lethal when they are infants. They could be die in the homozygous mice, and the heterozygous mice could survive.
Okay.
Yes.
So in the global knockout,
no difference. Yeah, yeah.
Answer two questions. Okay. So the first part of the question is related to what Clinton was talking about, why the animals body weight different, but the food intake about the same, whatever. I wonder, have you had a chance to do a metabolic profiling of those animals? That would allow you to tease out the intake and temperatures, especially you mentioned about adenosine involved in browning. If there's something to do with basal metabolic rate or thermogenesis, that could provide some answers, right?
Yeah, we have metabolic cage in the clipboard downstairs. Yeah, we can use that to conduct this kind of experiments. Yeah, thank you for your advice. Yeah.
My second part question is about your conclusion about adenosine sort of serving as a nutrient sensing and protective mechanism. So I was wondering, would you speculate, interpret, or some data show that adenosine expression, like increase with aging or decrease in those color-restricted, animals which is insulin sensitive?
Yeah.
If that's indeed the or you switch from high-fat diet to regular diet, would you see that level decrease? The reason I ask you that question is that because adenosine is known as a partial ligand of the genes we are interested in, ghrelin receptor. Ghrelin receptor we see in different tissues that change with age. And in the cells, there's the data show that adenosine and the ghrelin receptor GHS are from heterodimers. And in different tissues have some pretty interesting effect.
Okay. Thank you very much about this wonderful advice.
I've not advised them, just brainstorming.
Yep, the A2A adenosine receptor has high affinity to the adenosine, and as we know, there's 4 adenosine receptors. So when adenosine 2A is knocked out, maybe A2B adenosine receptor could compensate, but this has low affinity and is not as effective as A2AR. So it may compensate in some degree, but not as perfect like A2AR, the performance of A2AR. Yeah.
Okay. Well, if there's no more questions, join me in thanking y'all again for a great talk.